Hello and welcome to Johnson Biology News Network. Today I will be discussing the recent discoveries showing that there are tau accumulations in the brains of woodpeckers. Tau is a protein that builds up in the human brain due to trauma or old age. When in excess, tau can cause faulty communication between neurons. It can lead to cognitive, emotional, and motor malfunction, as well as resulting in neurodegenerative diseases such as Alzheimer's. It is also associated with the disease called chronic traumatic encephalopathy, or CTE. This is a disease normally seen in NFL football players and other athletes that experience repetitive, harmful head injuries. Previously, scientists had thought that woodpeckers avoid brain damage entirely when pecking due to an adaptation that allows their tongues to stretch as long as their wingspan. These tongues wrap around the brains of woodpeckers for extra cushioning, as pictured here. However, recent studies at Boston University have found data refuting the theory that woodpeckers do not experience brain damage. The woodpecker has more and more been referenced as a good model for protective sports equipment and protection against CTE in athletes, explaining why an in-depth and comprehensive study into woodpecker brain injury was warranted. The average human concussion occurs when impact of 60 to 100 G occurs, yet the woodpecker experiences forces between 1200 and 1400 G when their heads travel 15 miles an hour towards a tree and as many as 20 times a second, thousands of times a day. So essentially they are projecting their heads into a tree with 14 times the force of a human concussion just to eat. These are remarkable statistics that demonstrate the unique nature of woodpecker brains. An experiment was done where the brains of pileated woodpeckers were compared to red-winged blackbirds, which was the control. The pickled brains of deceased birds were sliced into thin segments and stained with molecules of silver, an element which attracts the protein tau. The data exhibited that none of the red-winged blackbird brains showed remnants of tau, whereas 4/5 of the woodpecker brains showed extreme amounts, including one adolescent bird. Considering the fact that woodpeckers experience no repercussions from such intense accumulation of the protein that causes aggression, memory loss, confusion, or depression in humans, shows that there are many differences in the framework of the brains of woodpeckers and humans. There are theories that the tau in woodpeckers could possess a predictive adaptation, giving hope to the idea that tau accumulation in humans and CTE conditions could be improved. Perhaps woodpeckers have more to teach us about brain functionality and CTE prevention, as research will continue to be conducted concerning this matter.
Good morning, I'm Polly, your news correspondent. We first begin with the news with a special report. Climate change has been one of the biggest topics so far in 2018. Currently, global warming and ocean acidification are contributing to a big problem among the marine food web systems by changing the energy flow to higher trophic levels. In addition to the alarming news, acidification can also cause rapid increase in cyanobacteria, which does not benefit the marine ecosystem. Disturbances in certain habitats and environments could cause the collapse of the marine food web system because certain species will not be able to adapt well. For example, the elevated temperatures can affect fish performance, such as increasing their metabolic rates and respiratory demands. These changes reduce their abilities to perform important life activities such as somatic growth and predator avoidance. The ability to forecast any predictions about how the food webs respond to climate change is limited due to the lack of knowledge about how individual species will react to the change themselves. A healthy marine food web has major importance since it affects human life and food production. If the climate continues to negatively impact the marine environment, this will majorly hurt many species on a global range. As a result of the climate changes, marine food webs will become more weakened in the future. Hopefully, with new findings and new technological advancements, the marine ecosystems will eventually improve. Meanwhile, here are some tips on how you can help these future problems. First, remember to go eat at your local restaurants and shop at your local grocery stores so that you can reduce your carbon footprint as much as possible. When going shopping, try to use a reusable bag or choose to have your groceries put in paper bags instead of plastic ones. Plan organizations or be part of a charity that helps our overall environment. Remember to recycle, pick up trash at your local parks and campsites, and use reusable water bottles instead of plastic ones. Last but not least, educate yourself on marine life and figure out what other things you can do to help save it. This has been Polly with the Morning Special Report. Have a nice day.
Norgard, your reporter at Johnson's Biology News Network. Now, everyone knows that the polar bear population is threatened. Whether you believe in global warming or not, the Arctic ice is melting, and with that, polar bears' habitats are getting smaller and smaller. However, a new study published by students and professors at the University of California, Santa Cruz in Science magazine suggests that the population of these mammals is declining faster than previously thought. Usually, as temperatures get warmer during the summer, the bears move inland and begin fasting until the ice returns in the fall season. However, ice in the Arctic is melting earlier each year and coming back even later, trapping the polar bears on land away from food for longer periods of time. Anthony Pagano, a PhD candidate, stated that this study identifies the mechanisms that are driving those declines by looking at the actual energy needs of polar bears and how often they're able to catch seals. Researchers on this project use collars with video cameras on them, like this one, along with metabolic tracers to observe hunting success, behavior, and the metabolic rates of 9 adult females with no cubs over an 8 to 11 day period. On average, polar bears spend about 50% of their time hunting and only actually catch something 2% of that time. They primarily use a hunting tactic called sit and wait, which, just like it sounds, means that the bears wait for seals to come to them before they attack them. Because of this, it was believed that they used a relatively small amount of energy to hunt compared to other animals. It was also assumed that the bears would use less energy in an unsuccessful attack than a successful one. However, this study revealed that the metabolic rates of these bears are actually 1.6 times greater than what was previously assumed, which means that they burn a lot more energy using the sit and wait tactic than what was believed. So polar bears are burning more energy and catching less food over the course of the year. Usually, bears with similar body masses to those studied need to, on average, eat 3 subadult seals every 10 days. However, the tested bears were only observed to eat 2 subadult seals every 10 days, which only allowed them to energetically break even, which means that they ate the bare minimum amount of food to survive. 5 of the 9 polar bears tested lost over 10% of their body mass over the 8 to 11 day period, meaning that they did not eat enough food to balance the energy they lost from hunting. These bears might be gone sooner than we thought, so we should do everything we can now to save them.
Hello, welcome to Johnson's Biology News Network. Scientists have recently discovered that those born with Down syndrome have an excellent genome to make up for the disabilities caused by the extra chromosome. Down syndrome is a genetic disorder also known as trisomy 21, where there is an extra 21st chromosome. This genetic abnormality is found in 1 out of 700 pregnancies, but only 20% survive the first trimester. Researchers have found that the genome of those with a disability is incredible, even better than those without. Geneticists tested gene variation, regulation, and expression of 380 individuals with Down syndrome and compared it to those without. The first test focused on rare variants that could be present, but researchers found that the mutations of the 3 chromosomes were identical and few in number, reducing variants. In the next step, it was discovered that people with Down syndrome had more regulators and diminished the expression, compensating for the extra chromosome. The final step was concentrated on the entire genome. The gene expressions were ranked on a scale of 0 to 100, with 50 being the ideal gene expression. Those without a disability had scored spread from 30 to 70, while those with Down syndrome were closer to 50 overall. This means the genome of someone with Down syndrome was more towards optimal and primary functioning. The geneticists concluded that in order for a fetus to survive the pregnancy, their genome must be of a higher quality to compensate for the disability. Finally, in this video, we see just how much Down syndrome is not scary, but more of an incredible feat.
Down syndrome is in your blood? Yeah. Does it make your blood special? Yeah. Is Down syndrome scary? No, it's not scary. It's so exciting. It's so exciting? Yeah. I get to go to Disneyland. You get to go to Disneyland? Yeah. We are going to Disneyland tomorrow. So Down syndrome is not something scary? No, it's not scary at all. It's not scary at all? No.
Good morning, and thank you for tuning in to the Johnson Biology News Network. I'm your host, Samson Thornhill, today with a news story about CRISPR. Sherlock is a new diagnostic tool invented using CRISPR technology to better help the health field in diagnosing illnesses. Sherlock stands for Specific High Sensitivity Reporter Unlocking, and they obviously chose the acronym so it can be Sherlock because it searches for viruses and then gives a detection based on that, even though the letters don't add up. Anyways, CRISPR, which is what Sherlock is based on, was originally designed to be used for gene editing. But the team that invented Sherlock, it can be used for gene detection instead of for gene editing. The way Sherlock works is this. CRISPR has a main protein called CAS, and then there's a bunch of different variations of that. In Sherlock's case, they use Cas13, and it's programmed in the lab prior to being given to detect for a certain strand of RNA. For example, some Sherlock tests might be programmed to detect Zika, while others might be programmed to detect dengue virus, both causing similar physical symptoms in patients, making them hard to detect. Upon the Sherlock test being placed into a solution, CAS13 will search for the RNA strand, chop it off, and release some signaling molecule to transfer information to the test. Physically speaking, the test is a strip of paper that will display a line or lines to signify a specific result, similar to a pregnancy test. According to Jonathan Gutenberg, a Harvard graduate and co-author for the paper detailing these feats, the new paper readout for Sherlock lets you see whether your target was present in the sample without instrumentation. This makes previously complex and expensive testing for diseases far simpler and more cost-effective, mostly simply requiring just a blood sample and a few minutes. Sherlock has been revamped by this team to be even more efficient, sensitive, and powerful. In fact, Sherlock can now detect up to 4 different molecules in one sample, and it's over 100 times more effective. Additionally, it displays results very quickly, making long and painful tests that leave patients wondering for days obsolete. If Sherlock were to be mainstreamed, the medical field would change forever because of how affordable testing could become. Thank you.
Good morning and welcome to Johnson Biology News Network. I'm your host, Julian Anderson, and today I come to you with a striking new discovery in the science world. A study from the Laboratory of Sensory Motor Research at the National Eye Institute emerged 1 week ago, the 15th of February, in which visual selective attention was studied in mice. Despite non-human primates, such as monkeys and apes, being the primary animal model for studying visual perception due to their similarity to humans, scientists have found that mice, yes, mice, are also valuable to such studies due to their molecular and genetic tools. Prior to the study, mice had not yet demonstrated visual selective attention capabilities. Visual selective attention is defined as the ability to limit neural processing and behavior to relevant signals while actively ignoring other irrelevant or distracting signals. In the experiment, a mouse was placed on a running wheel with its head restrained by a head post. In front of the mouse were 2 displays on both the left and the right side, and directly in front of the mouse was a lick spout. The mice had been trained to lick the lick spout when they detect a change in the visual display. On either or both visual displays, a Gabor patch was shown, as above. As soon as the patch changed orientation, mice were expected to lick the spout within 500 milliseconds or half of a second. 3 different experimental versions took place demonstrating 3 different paradigms. Experiment 1 used a Posner-style cue, which indicated the probable location of the relevant visual event. The results demonstrated that accuracy was higher and reaction times with the tongue were shorter with a cue present. Experiment 2 presented a cue versus no cue task, where the cue was only present on half of the trials. The results suggest that mice had higher accuracy and lower reaction times with spatial cues compared to trials where the cue was not present. In experiment 3, the spatial cue indicated the location of the relevant visual event. Researchers found that mice could be trained to ignore identical yet irrelevant visual events at uncued locations. So what does all this research mean? Because of the discovery that mice can use visual selective attention skills, mice have been established as a valid animal model for future studies of the genetic and neural circuit mechanisms of visual selective attention. But it also means that your parents are probably correct when they accuse you of selectively ignoring them. Julian Anderson, Johnson Biology News Network.
Hi, my name is Andrea Rodas, and welcome back to Johnson Biology News Network. For today's topic, there has been a recent discovery involving the human brain and musical rhythms. In an experiment led by Jessica Ross, 25 healthy adults underwent continuous theta burst simulations applied to 2 areas of the brain. The first would be the supplementary motor area, highlighted in red in the picture here, and the posterior parietal cortex, highlighted in blue, also known as the IPVC. Continuous theta burst simulations are magnetic pulses which were used to intentionally disable the functions of those parts of the brains for about an hour. After being suppressed, the adults were tested on their abilities on recognizing tempo changes, timing intervals, and changes in rhythm. There were no significant findings involving the supplementary motor area. However, the test subjects struggled to identify rhythm changes when the posterior parietal cortex was suppressed. This indicates a relationship between the dorsal auditory system, to which the IPVC belongs, and the recognition of rhythm. So next time you feel the need to move to the beat, blame it on your posterior parietal cortex. See you next time on Johnson Biology News.
Good morning, my name is Jbnn and welcome to Johnson Biology News Network. Today I'll be talking about genetics and how they can be used in forensics to identify time of death. Computational biologist Roderick Guigo of the Center for Genomic Regulation in Barcelona, Spain, is part of the Genomic Tissue Expression Pilot, a large group of geneticists and molecular biologists that have been measuring gene activity in tissues from hundreds of people, living and dead. The goal is to determine how the body makes different cells to do different things, given that they all carry the same DNA instructions. It also seeks to determine how slight variations in DNA from person to person change what cells do. Other researchers have already shown that some genes stay active up to 4 days after death. Guigo wanted to find out how gene activity changes as the time of preservation is extended. The response to the death of the organism is quite tissue specific. For example, there were very few changes over time in the brain's or spleen's gene activity. More than 600 muscle genes either quickly increase or decrease activity after the loss of life. Guigo and his colleagues developed a software that learned the patterns of 399 people and then tested how well the machine learning software did predicting the time of death of 129 other people. The software discovered, for example, that in blood, decreased activity of genes involved in DNA production, immune response, and metabolism, but an increase in those involved with stress responses signaled the person had died about 6 hours before preservation. The majority of gene activity changes, both increases and decreases, occur between 7 and 14 hours after death. Then after 14 hours, gene activity seems to stabilize. This software is the first step towards harnessing gene activity for forensics, although Guigo's group has shown that the time of death can be estimated just as well using gene activity levels from 2 tissues, the lung and the thyroid. His team have not yet been able to reduce the number of genes needed to make the prediction. The more genes analyzed, the more expensive the work. Even so, Guigo is eager to see what else he can learn from these patterns. Changes in gene expression may also carry the signatures of the cause of death, he says. They don't have enough detailed information to investigate the hypothesis. Perhaps they're saving this for future studies.
Hello, and welcome to Johnson's Biology News Network. I'm your host, Katie McCrum, here with today's most recent updates. In this segment, we will be discussing the recently discovered biomarkers of autism spectrum disorder, which are based upon symptom severity and its measurements. Facing our community today is the enigmatic disorder labeled as autism spectrum disorder. The main issue with it is that no physicians are keenly aware of its causes, therefore indicating much difficulty when it comes to learning how to diagnose it. Today's breaking news story regarding this disorder has enumerated the details regarding the research of blood biomarkers and their effects on finding the biological signs of autism spectrum disorder, possibly even before its symptoms appear. The unknown causes of autism spectrum disorder are a pressing issue in today's world of biology, medicine, and the daily lives of those who autism affects. The ambiguity of what causes the disorder makes the general population not only concerned but also cautious. This cutting-edge research provides a wealth of knowledge for those who are affected by autism spectrum disorder in any way. The disorder's symptoms are measured and ranked by severity and how pronounced the typical autistic behaviors are. In this recent discovery, measurements were recorded on the basis of social impairments, repetitive behaviors, stereotype behaviors, and the correlation of neuropeptides and their receptors. The biomarker presence is a definitive quality in determining whether or not an individual has autism spectrum disorder or will likely develop it in the coming years. Found in the blood, each of the neuropeptide biomarkers dictate the levels of oxytocin and arginine vasopressin and their respective receptors, and they represent the likelihood of a child having the disorder. Oxytocin and arginine vasopressin are strong contributors to the social behaviors and the ability to determine emotions, the key external markers of autism spectrum disorder. SRS tests were given to the children diagnosed with autism spectrum disorder and indicated a strong correlation between their symptoms and actions, habits, and emotions in social situations, what the test was designed to measure. The SRS was given as a precursor to the blood test to determine which group each individual would be categorized into. These test results were then followed up with a uniform blood drawing to measure the levels of the neuropeptides oxytocin and arginine vasopressin. The blood drawing was conducted in a succinct manner, and as the experts continued to find the concentration of neuropeptide levels to then evaluate the gene expression levels as well as its sequencing. This was highly important to the process in order to make further advancements to achieve the discovery of autism spectrum disorder's causes. As a result of measuring the neuropeptide concentrations, it was concluded that the children's social behaviors and functions did not correlate with the neuropeptide level. In contrast, the low neuropeptide concentrations indicated a higher severity of symptoms, especially those highlighted in the SRS. Intellectual impairment was not a factor in neuropeptide concentrations due to the characterization of autism spectrum disorder. Lastly, the trial directly showed the gene expression levels and how they were affected by the biomarkers found in the blood, which also correlated with the neuropeptide receptors. In summary, the results of the experiment indicated a relation between the neuropeptide concentrations in the blood along with a simple blood drawing. The blood should then be followed with a test to determine the neuropeptide concentrations as they correlate with the onset symptoms. Finally, if the neuropeptide levels are lower than average, then the probability of the individual's autism spectrum disorder diagnosis is highly likely. This new advancement will not only work in the lives of those with the disorder, but will affect the world of medicine in all future diagnosis endeavors. This has been Katie McCrum of Johnson's Biology News Network with today's breaking news story. Thanks for watching.
Good morning. I'm Zoriz Azam at Johnson's Biology News Network. The cheetah is the world's fastest land animal that is successful at hunting, not only because of its rapid speed, but also because of its incredible still gaze while chasing prey. Researchers investigated for the first time the cheetah's remarkably sensory abilities by examining the inner ear. The cheetah's inner ear is an organ that is necessary for maintaining body balance and adapting head posture. Researchers of the field find the inner ear of cheetahs is distinctive and likely evolved recently. When a cheetah is in motion, its legs and back are moving incredibly fast, but the head barely moves at all. The inner ear makes it easier for the cheetah to maintain visual and postural stability while running and chasing its prey. No one has investigated the inner ear's role in cheetahs until now. The inner ear is made up of 3 semicircular canals that carry fluid and sensory hair cells that detect movement of the head. These semicircular canals are positioned at different angles and are sensitive to different movements such as up and down or side-to-side movement. Researchers use X-ray computed tomography to scan the skulls of 21 felid specimens. Those include modern cheetahs from distinct population, a closely related extinct cheetah, and many other living felid species. With the data, researchers created detailed 3D virtual images of inner ear shape and dimensions of each species. They found that the inner ears of living cheetahs differ significantly from the inner ears of all others found alive today. The inner ears of the cheetahs had a great volume of vestibular system and longer anterior and posterior semicircular canals. The distinctive inner ear of a cheetah shows enhanced sensitivity and more rapid responses to head motions, which explains the animal's stunning ability to keep its vision stable and to maintain its gaze still on its prey when running at high speed. The extinct species did not have these traits as studied by researchers. This emphasizes the recent evolution of the inner ear of modern cheetahs. By using high-tech equipment to look at the inside of the skulls of modern cat species, researchers have discovered that there are unique movement and sensory system adaptations to high-speed predation in the cheetah ancestry. The competition with other predators has probably forced cheetahs to develop a high-speed hunting strategy. The cheetah's ancestors were able to run very fast, and then the inner ear easily affected the head movements to keep the head still, allowing them to run even faster.
Hello, and thank you for watching Johnson's Biology News Network. I'm your host, Riley Myers, and today we'll be reviewing a newly released article from Current Biology. In this article, scientists have discovered a unique way of analyzing how the female zebra finches recognize courtship calls. In general, birds use vocal cues in order to communicate information regarding the caller's identity and worthiness. Females can recognize the differences between these calls, and it allows them to understand whether they are courtship calls or general social interaction calls. In the past, scientists have thoroughly studied auditory preferences but have not dug deeper. In this study, however, scientists actually analyzed the brain and how regions are selectively activated by female-directed songs. A functional MRI was used to evaluate the activation of different parts of the brain to these courtship calls. By looking at the entire avian brain, the MRI localized the activation to 2 specific regions. This data was then confirmed by immunocytochemistry. The 2 identified regions were the caudocentral nidopolym, or NCC, and the caudomedical mesopolym, or CMM. The CMM was established as the part of the brain that is involved in the actual interpretation of the mating call, whereas the NCC is more directly linked to how the finch responds to the call. The article discusses at length how these 2 regions were identified. They were isolated and tested upon. Furthermore, these findings conclude that these 2 regions are involved, but provides no evidence that these are the only 2 regions that are involved. We are only beginning to scratch the surface of exploring the neurological interpretation of bird mating calls. And then this is the call. Thank you for joining, and catch us next time on Johnson's Biology News Network.
Good morning, and welcome to Johnson's Biology News Network. Over the past few decades, rapid industrialization, increases in population density, and changes in agricultural practices have contributed significantly to the contamination of bodies of water with hazardous pollutants. Specific industries such as mining, smelting, refining, and battery manufacturing release large amounts of heavy metals including lead, chromium, mercury, and arsenic. Pollution caused by heavy metals are a major concern as they are resistant to degeneration and cause morphological as well as physiological damages to aquatic fauna. The introduction of heavy metals into fish populations is frightening, as fish serve an important role in the aquatic food chain and are a main part of the diet of humans worldwide. In a recent experiment conducted by Indian researchers, fingerlings, in other words baby fish, were tested in baths of lead-polluted water in order to observe the mortality rate in a toxic environment. The researchers chose to perform their experiment using Margala carp, scientifically known as Sarhinus margala, as it is one of the 3 major Indian carps and is cultivated throughout southern Asia. The fingerlings were acquired through a local fish hatchery and given a week to adjust to laboratory conditions. They were placed in plastic troughs at 28 degrees Celsius with 10 fingerlings per trough. Lead nitrate serves as the heavy metal in this experiment and was prepared in solutions with varying concentration. The concentrations of lead nitrate range from 1 parts per million to 500 parts per million. A plastic trough containing only unchlorinated water served as the control. The fish were then kept in the troughs for a period of 96 hours and were continued to be fed twice a day. After a 96-hour period, the number of dead fish was recorded. During the experiment, it was observed that the fingerlings in concentrations of lead nitrate above 40 parts per million surfaced more frequently, indicating that the fishes were showing increasing degrees of anoxia with increasing concentrations of lead nitrate. It was noted further that the irritability of the fingerlings increased considerably when exposed to the toxic environment. The body of the fingerlings became covered by an opaque film, which looked like a coagulated mucus. The thickness of the film increased proportionally with the increase in the concentration of the lead nitrate in the experimental solution. The gills were also covered by a film of similar nature but thicker. When examined under a simple microscope, degenerative changes to the gills were also noted. The movement of the fishes exposed to higher concentration of lead nitrate became sluggish and eventually losing equilibrium with death following soon afterwards. The study shows that Margala carp fingerlings could survive the toxic effects of lead nitrate up to 40 parts per million without any physiological stress. Above 40 parts per million, the physiological stress sets in as indicated by increasing surfacing of fingerlings. In addition, the gills of the fingerlings display a high degree of dysfunction with increasing concentration of the lead nitrate. Death seemed to be caused by asphyxiation due to the reaction of heavy metal ions on the mucus secreted by the gills. It had been shown earlier that the primary effects of lead nitrate is the prevention of the diffusion of oxygen into the gill capillaries, leading to anoxia. Thus, apart from the interface of lead ions with the fingerlings' homeostatic systems, physical obstruction to oxygen diffusion also occurs. Lastly, the lead nitrate did not cause mortality at sublethal concentrations. Prolonged exposure can lead to undesirable effects like a shorter lifespan. The evidence produced by the study is shocking and disturbing, as it demonstrates that fish, even with a substantial concentration of heavy metal within them, are capable of surviving. This may prove fatal to humans or other animals eating these fish to survive. In order to stay safe from any lead or heavy metal poisoning, make sure to always check where the fish you eat are sourced and whether or not there are any fish consumption advisories in your local area. This has been Yash Corday for Johnson's Biology News Network, wishing you a great and science-filled day.
Hi, Alex Buchanan here for Johnson Biology News Network, where we keep you updated with the latest and greatest advancements in the field. In the world of cell biology, cancer research often claims the center of attention, and justly so as the disease is becoming more and more common. Today, we address a study on precancerous tumors. Within tumors, there are often hypoxic microenvironments. In these low oxygen areas, cells of course cannot function as they normally would. In this state, they produce hypoxia-inducible factor 1 alpha, also known as HIF-1 alpha. This protein activates a transcription of a number of essential genes for survival. one of which is vascularization. It's extremely vital in a low oxygen situation, and because of this, cells can receive more blood and in many cases move away from their hypoxic state and back into an oxygen-rich state. Despite how crucial HIF-1 alpha is, for reasons that we've yet to discover, it seems that cells do not always make enough of this protein for the cell to return to normal, or for it even to function correctly in a low oxygen environment. As if these problems weren't bad enough, low oxygen areas are also understood to be niches for stem cells to develop. So when these irregularities come up, cells often don't even respond with self-destruction, because in hypoxic environments, HI1 alpha is also responsible for signaling the transcription of the gene for apoptosis. Cells acquire this necessary information about their surroundings with strings of monosaccharides called glycans on the outside of their cell membrane. The final carbohydrate on the chain is often the most important. Just 6 days ago, a study was released in which Robert B. Jones led a team reviewing the correlation of ST6-Gal1, a mediator for placing this crucial final carbohydrate, and HIF-1 alpha. The team found that cells completely grown in hypoxia see more use out of ST6-Gal1, and they even suggested that such changed intracellular to extracellular connections, and thus somehow led to a greater expression of HIF-1 alpha. This is one of the first studies of its kind, but if this knowledge could be used to intentionally increase the expression of HIF-1 alpha, more tumor cells could function in a normal way and begin apoptosis if necessary. Stem cells would be likely to develop, less likely to develop, and cancer could even be prevented before it ever started. This is Alex Buchanan, signing off for Johnson Biology News Network.
Hello, and welcome to the Johnson Biology News Network. I'm your host, Sophia Parrott, and today we will be diving into the recent discoveries of how climate change affects Mediterranean high mountain plants. Many high mountain zones are ideal environments to study climate change as they host diversity and sensitivity, and there is less human interference than on lowlands. Key characteristics of Mediterranean mountains, however, is that warming generally leads to a reduction in precipitation, declines in spring snowpack, and an increase of extreme events such as drought. This makes Mediterranean mountain ranges particularly sensitive to climate change, which is why much of the study correlates to this specific range. Prior studies suggest that affected plants all share the same level of and types of risks when encountering climate change. However, recent studies more accurately pinpoint exactly what areas of the mountain range experience a certain type of risk. SDMs, or species distribution models, projected in previous studies were also proven inaccurate, as they did not consider the phenotypic plasticity, adaptive potential, dispersal efficiency, or species interactions of the plants. Essentially, the more recent investigation indicates that the effects of climate change on highland plants largely depend on what area of steep mountain slope they are located on. Surveys on the Mediterranean high mountains in particular confirm an upward shift of drought-tolerant lowland plants, which is associated with a decline in the number of highland native species. This is due to climate change, which causes decreases in precipitation and snowfall and increases in droughts. Since high mountain plants rely on the cold for survival, they begin to move down where less droughts are prevalent, and lowland plants move up. Climate change can affect high mountain plants in a variety of ways. It may influence plant phenology and reproductive success, since the flowering cycle in alpine ecosystems is driven by cold weather. Warming can therefore cause these plants to respond by flowering at unfavorable growing seasons instead of flowering at their usual time and pace. Drought and warming therefore have negative effects on flower and food production. Specific reproductive risks include pollinator limitation. Climate trends may alter the abundance and distribution of pollinators, causing phenological mismatches and plants' inability to receive the aid they need to reproduce. Here we see two types of the most important pollinator groups of Mediterranean high mountain plants. Butterflies are particularly sensitive to climate change, and this is the Apollo butterfly, or the mountain butterfly. Climate changes also affect post-pollination processes in which seed germination is never triggered due to inadequate precipitation or weather and plant population dynamics. The good news is that these high mountain plants adapt so as to extend growth rates and compensate for the negative effects of fluctuating climate trends. The bad news is the rate at which these plants adapt are much too slow in comparison to the rate of climate change. In response to this dilemma, researchers in this study offer suggestions for future observations. They recommend comparative studies between taxa or populations of different biomes, long-term studies and experimental manipulations, studies in demographic changes, garden and reciprocal translocation studies, and studies in plant-to-animal and plant-to-plant relationships. Hopefully, these future investigations will help provide solutions to the demographic decline and topographic shifts of Mediterranean high mountain plants. This has been Sophia Peck on Johnson Biology News Network. Thank you.
Hello, and welcome to Johnson's Biology News Network. Today we will be discussing a recent study conducted in 2017 during which discoveries were made about the effects of preterm birth and very low birth weight infants, or VLBW births, on cognitive, behavioral, and emotional problems in later life. A VLBW is defined as weighing less than 1,500 pounds or 3.31 pounds at birth. These infants are at higher risk of perinatal brain injuries and unusual or delayed brain development. Both hyper and hypoactivation of the brain are observed more commonly in low birth weight adults based on bold fMRI scanning, which can be linked to Alzheimer's disease in later life. Deficiencies in stable task maintenance and adaptive task control have been exhibited frequently through VLBW adults, meaning that both brain processing over an extended period of time and rapid momentary processing may show delay. Though alterations in brain structure and decreased neurodevelopment are clear in those born preterm or at low birth weight, functional adaptations are more difficult to examine. Throughout the study, 32 adults born preterm and at very low birth weight were compared to the control group of 32 term-born and average-weight adults. To score the individual's cognitive control, the time it took for them to complete the Dellis-Kaplan Executive Function System Trail-Making Test was recorded. Fluid intelligence of the subjects was based on the Welsher Adult Intelligence Scale 3, and anxiety was measured using the Adult Self-Reported Anxiety Scale of the Achenbach System of Empirically Based Assessment. There was no statistical significance supporting the increased self-reported anxiety levels of the experimental group, though they did exhibit a lower fluid intelligence score than the control group. The projected image shows the results of the fMRI, illustrating the cognitive control of the participants. The stable task maintenance, or STM, results show increased activation in the control group, which focus on two significant clusters, one in the region of the frontal pole and the anterior cingulate gyrus, and the other in the posterior cingulate gyrus and the precuneus. The ATC, or adaptive task control, gave opposite results in that there were increased activations in the experimental group with significant clusters focused in regions in the posterior cingulate gyrus and the precuneus, and in the right lateral occipital cortex and angular gyrus. The threshold PMAP shows blue areas which are not significantly different between the two groups based on observed data. The green and red areas, however, indicate the VLBW individuals have a lower fractional anisotropy, reflecting density of fibers and myelination of white matter. Their mean diffusivity, however, is greater than that of the control group. Bold fMRI activations were used to compare gestational age, birth weight, and task performance through the regions of interest of the brain. The major conclusions of the study state that the VLBW population demonstrates deviant hemodynamic responses in correlation with the reactive cognitive control. The hyperreactivity also exhibited a lesser white matter organization in the brains of those born at lower gestational age and birth weight. The research provides evidence that supports the claim made by Alexander Olson that suboptimal organization in the central nervous system due to the consequences of preterm birth with VLBW leads to a dysfunctional hyperreactive brain state that extends into adulthood.
Hello and welcome to Johnson Biology News Network. I'm your host Zoe Bell, and today we'll be talking about dolphins. Dolphins use echolocation to sense their surroundings. They do this by clicking at high frequencies. This high frequency, called sound waves, travels through the water to an object and then back to the dolphin. This dolphin clicks by having air pushed through their nasal passage along with their phonic lips. The tissue around them vibrates, which creates clicking sounds, which then creates a sound wave. Different species of dolphins generate different clicking noises. According to a recent development, there is a new algorithm that distinguishes different species of dolphins by the way they use this echolocation. This algorithm was originally created to figure out how the species of dolphins as a whole were affected by various harmful activities in the Gulf of Mexico. Examples of this include the 2010 Deepwater Horizon oil spill, climate changes, and various natural resource developments. It was difficult to differentiate the dolphin clicks, so Caitlin Fraser of Scripps Institution of Oceanography, California, and her colleagues created this algorithm to attempt to distinguish the different clicks of each dolphin in said area. In addition to differentiating the dolphin clicks of each dolphin species, the algorithm was also an advancement in the constant tracking of the location of dolphins. Before this algorithm, tracking the location of a dolphin was based on what people saw rather than any technology, and unsupervised technology at that. The algorithm is a huge improvement in many ways. It is safer for the dolphin because the new technology has proven to be non-invasive. It is unsupervised so that there is less human error. And best of all, scientists can now distinguish different dolphins and track them for years at a time. And then here's a video clip showing the dolphin. Clicking off.
Hello, and welcome back to Johnson's Biology News Network. I'm your host, Amber Adams, here to bring you a special report in neuroscience. Recent discoveries have found that dopamine neurons are a possible factor in altering one's biological clock regarding the sleep-wake cycles of circadian rhythm. A team of researchers from the University of Virginia examined the neurological underpinnings of a human's internal clock. Located throughout the body are different groups of interacting molecules that are connected by the center. The suprachiasmatic nucleus, or SCN, consists of more than 20,000 neurons and is found in the brain's hypothalamus. This main master clock regulates each individual group of interacting molecules. According to the new study published by the Journal of Current Biology, dopamine-producing neurons have been found to directly connect to the brain's SCN. Dopamine is a neurotransmitter with a variety of functions, such as controlling pleasure, signaling, learning, and movement. Moreover, this compound aids the brain in processing rewards, as well as detects for available food or partners. The ventral tegmental area is the origin of dopaminergic cell bodies of the mesocortical dopamine system and other dopamine pathways. The amount of or absence of light is an external stimuli which also regulates the activity of the biological clock. Ali Denise Gieler, a University of Virginia professor of biology and neuroscience, hosted a study regarding this research in his laboratory. Ryan Grippo, a PhD candidate and a student of the professor, led a team in the examination of two groups of mice. In the experimental group, the dopamine receptor signaling within the SCN was genetically knocked off in order for the mice to have a disrupted dopamine circuit. This caused the communication between dopamine neurons in the VTA and the SCN to cease. The other group of mice was kept under normal conditions to serve as the control group. In order to mimic conditions of jet lag, the researchers altered the light schedules of both groups by 6 hours. The researchers observed that the genetically modified group of mice adapted to the schedule shift comparatively slower to the control group. This supported the hypothesis that dopamine signaling between the VTA and the SCN is needed to regulate circadian rhythm. Regarding the difficulty in retiring early to bed, Professor Gieler explains how engaging in rewarding activities, such as eating or watching television, actually inadvertently affects our biological rhythms. Moreover, he commented that discovering how the dopaminergic and circadian rhythm systems connect together further aids in targeting these neurons with therapies that have the potential to provide relief of symptoms, specifically for travelers and shift workers, and possibly for people with insomnia. Neuroscientists now believe they possess novel insight into how dopaminergic pathologies, including Parkinson's disease and ADHD, affect circadian systems behaviors, such as sleeping. Stay tuned for the next discovery in biology. This is Amber Adams signing off.
Hello and welcome to Johnson News Network. I'm your host, Bryce Brooks, here to bring you the news on the newest biological discoveries. Recently, scientists have observed how the Feronia receptor kinase maintains cell wall integrity during salt stress through calcium signaling. Feronia, a plasma membrane localized receptor kinase from Arabidopsis, shows its purpose and need by assisting the ground root back into recovery and growth when exposed to high salinity. Salinity regarding the salt content found in the soil or solution. High levels of salinity can be found in both natural and agricultural environments and impose osmotic and ionic stress, limiting the capability of cells to intake water. The domain of Feronia shares many qualities with that of malactin, an animal protein for binding diglucose with a purpose for protein quality control in endoplasmic reticulum. With an extracellular domain similar to malactin, it is now thought that Feronia, as well as other related receptor kinases, through interaction with cell wall polysaccharides, pose direct correlation to a wall sensing function. High salinity in soil poses a threat by softening the cell wall, and when disrupted by the Feronia mutant, the cells result in an explosion of growth recovery. Additionally, when two independent mutant alleles of MURAS1 were introduced, the cell wall integrity resulted in a burst of cell growth, a similar-like effect to Feronia, however much weaker. Furthermore, the two independent mutant alleles disrupt pectin cross-linking. As for Feronia, with the treatment of calcium and borate, pectin cross-linking is facilitated. With a sense of these wall defects caused by salinity, a direct consequence of the extracellular domain of Feronia, and pectin might therefore be resulted. Feronia-dependent signaling allows for cell-specific calcium transients to uphold cell wall integrity during salt stress. This shows the correlation of Feronia as a sensor of damage to pectin-associated wall. Overall, Feronia is required for root growth recovery during high salinity. Here in Figure A, seedlings of Arabidopsis WT Ferona 4 and Ferona complemented line 24 hours after transfer to the media. The black lines mark the position of the root tip at the time of transfer. And B, root growth for WT, 2 Ferona alleles and 2 independent Ferona complemented lines 24 hours after transfer to media. And C, quantification of hourly growth rate of WT and Ferona 4 roots transferred to media, containing 140 millimeters of NaCl. And D, root growth of WT, Ferona 4, Ralph 2, Ralph 1, RNAi, and Ralph 1 overexpression lines 24 hours after transfer to media. That covers all in the new biological discoveries. Thanks for tuning in. This has been Bryce Burks.
Welcome to Johnson Biology News Network. I'm your host Cameron Chandler, and today we'll be discussing the cerebrovascular pathology in individuals with Down syndrome and Alzheimer's disease. Researchers at the University of California at Irvine Alzheimer's Disease Research Center have examined how cerebrovascular amyloid angiopathy affects individuals with Down syndrome and individuals with Alzheimer's differently. 3 groups of deceased individuals were examined: individuals with Down syndrome, individuals with Alzheimer's disease, and non-demented individuals were used as a control. It's commonly known that individuals with Down syndrome suffer from memory loss earlier in life and more severely than non-demented individuals. Researchers hypothesized that the individuals with Down syndrome would have cerebral amyloid angiopathy worse than the other individuals because they have a 3rd copy of the 21st chromosome, which has a gene that codes for amyloid precursor protein. The amyloid precursor protein leads to overproduction of amyloid plaques, which leads to memory loss. The cerebrovascular pathology was examined by looking at amyloid stains, the circle of Willis, and other blood vessels in the thalamus and inside the brain. Arteriolosclerosis was interesting because the thalamus and many parts of the brain had to be looked at. The findings were in line with what the researchers had expected, which is that the Down syndrome individuals had a lot of cerebral amyloid angiopathy, 88% of them. 33% were severe, which is over double of those with just Alzheimer's disease. Trisomy 21 caused that overproduction, and the CAA also worsened with age, but in individuals with only a partial trisomy of the 21st chromosome didn't have the neuropathy for Alzheimer's disease. So it's strongly believed that the overproduction is only caused by that gene. For future research into the memory loss that individuals with Down syndrome face, it's critical to also investigate the cerebral amyloid angiopathy, as it's critical to how the plaques block their blood vessels. Thank you.
Hello, and welcome to the Johnson Biology News Network. I'm your host, Julia Wolfer, and today we will be discussing genome editing in stem cells for disease therapeutics. Genome editing is the process of inserting, deleting, or replacing a section or sections of DNA in a target cell. 3 generations of programmable nuclease systems can control editing at a specific site. The first generation are zinc finger nucleases, or ZFNs for short. They are the smallest and contain an FOKI nuclease domain, and a single subunit binds to 9 to 18 base pairs of the DNA sequence. Disadvantages of ZFNs include high rates of off-target effects and cytotoxicity. The second generation are transcription activator-like effector nucleases, or TALENs for short. They also contain an FOKI nuclease domain, but have a repeating unit of 33 to 35 amino acid repeats. They are designed in pairs that bind opposite DNA-specific sites. Advantages of TALENs include high efficiency with relatively low rates of off-target effects and cytotoxicity. The third generation are clustered, regularly interspaced short palindrome repeats, also called the CRISPR/Cas9 system, which is composed of a single guide RNA and a specific endonuclease protein, which is the Cas9. This system requires a protospacer-associated motif, or PAM for short, that matches the RNA sequence. This nuclease system is more easily designed and applied to any genome sequence than ZFNs or TALENs. These programmable nuclease systems can be used to reprogram induced pluripotent stem cells with transcription factors from somatic cells for disease modeling and developing therapeutics. Genome editing can be used to generate or correct stem cell-based disease models by either introducing the disease-causing mutation into healthy control stem cells or correcting the disease-causing mutation in patient stem cells. Cells that have been edited ex vivo can be transplanted and reintroduced back into the patient. ZFN-based genome editing with stem cells has been used to generate disease models and to develop cell-based therapeutics. Neurodegenerative models, including Parkinson's disease, Down syndrome, and amyotrophic lateral sclerosis, also known as ALS, have been studied using this. TALENs can be used to generate various genetic diseases in order to investigate the mechanisms by which mutations occur. A specific instance is in presenilin-1 and how mutations contribute to familial Alzheimer's disease. In one study, intestinal stem cells derived from patients with cystic fibrosis were corrected with CRISPR/Cas9. Corrected cells were shown to be fully functional. Intestinal stem cells and CRISPR/Cas9 have also been applied to colon cancer modeling by mutating the cancer-related genes to analyze the involvement of each gene in colon cancer pathways. The discovery and characterization of the programmable nucleases used in ZFNs, TALENs, and CRISPR/Cas9 systems have great potential for the future of technological advancement in genetic therapeutics. While the application is significant, guidelines must also be carefully planned in the future to ensure a balance between the enormous possibilities of these tools and the risk of their misuse. This has been Julia Wolfer at the Johnson Biology News Network. Thank you for watching.
Hello, welcome to Johnson's Biology News Network. I'm your host, Logan Stevenson, and today we have breaking news: Fly Fight Night. The recent study investigates whether aggressive encounters where Drosophila melanogaster males adopt a winner or loser state have long-term effects on qualities of persistence, internal state, and behavior in aspects other than fighting. First, to investigate the effect of fighting experience on behavior, the lab team had to create chronic winners and losers. All the flies were trained in 3 30-minute time periods with an hour break in between. To do this, one group of flies was paired with hypoaggressive flies that executed around 10 lunges in 30 minutes. These flies would be the chronic winners, as they dominated the hypoaggressive flies. The second group would be paired with hyperaggressive flies, which executed around 210 lunges in 30 minutes. These flies would become the chronic losers and often tried to escape the training area. To test whether successive rounds of winning or losing created long-lasting internal states, they paired both winners and losers with a normal fly at 24 hours, 48 hours, and 72 hours after training. As shown in the charts, winners executed significantly more lunges than their opponents in all time periods after. Similarly, the losers significantly received more lunges than their opponents in the 24-hour and 48-hour time periods. These results support that aggressive encounters do have long-lasting effects on internal state and persistence. Additionally, they tested to see whether flies associated the fights as negative or positive. They did this by exposing the winners and losers first to odor 1, associated with no opponent, then odor 2, in the presence of an opponent. After 24 hours, it was shown that the winners had an attraction to odor 2, while the losers had an aversion to odor 2, thus illustrating that winning is perceived as rewarding while losing is seen as aversive. Furthermore, the lab tested whether the aggressive encounters affected other behaviors. The first test was created to test the risk-taking of winners and losers. In this, flies whose wings had been removed were given 10 trials to cross a 3-millimeter gap. Normal flies, or the control flies, had a 50% success rate. The winner fly group showed a significantly higher rate of successful crosses, while the loser group had a significantly lower success rate, as shown in the first chart. Thus, the researchers support the idea that prior fighting experience affects central sensory processing such that winners are more willing to cross the gap than losers. Next, the scientists investigated winning and losing on courtship behavior. To test this, they compared the fly's time to display the first wing extension, as well as the total time performing courtship behavior and mating. It was shown that the loser took longer than the winner in total time courting and mating with the female flies, while winners showed their wing extensions quicker, thus initiating the start of the mating ritual faster than the losers. And this is shown in the second graph. These results show that loser flies have a lower drive to court females than winners. Finally, the last test was based on winner and losers flies exploration. Flies tend to be attracted to tall objects. Thus, to test the exploration of the flies, the experimenter observed whether the flies went towards the object and how long it took. In the study, they found that both winner and loser flies went to the object. However, the winner flies reached the object on average 200 seconds faster. In conclusion, these experiments show that the aggressive encounters of flies can have long-term effects on their internal states, associating winning as rewarding and losing as aversive. These effects on internal state then create generalizations that can influence fly behavior in other areas. Thank you for watching.
Hello everyone, welcome to Johnson Biology News Network. I'm your host, Charlie Moose, bringing you the latest on biological news. The latest story out of Australia concerns the curiously unique biological traits of the duck-billed platypus. Platypi, part of the monotreme family, have remained an enigma in the biological world for their strange traits and appearance. They are beaver-tailed, duck-billed, webbed-footed, venomous, egg-laying mammals, basically the perfect subject for unique evolutionary research. A team of researchers decided to look into platypi's strange method of nursing their young. As they have no external mammary glands, they provide milk for their young by exuding the breast milk onto their bellies and having their young lap it up. This technique does allow their young to nurse, but it also exposes them to harmful bacteria. Since the milk is lapped up off of the belly, any dirt or bacteria on the mother's skin could contaminate the breast milk. However, the research team discovered how their young can be so vulnerable, yet remain so healthy. After examination of a mother's breast milk, the team found a never-before-seen protein. The protein, dubbed the monotreme lactation protein, or MLP for short, was synthetically produced in a lab to further examine its structure and use. While all species of mammals' breast milk include some type of antimicrobial proteins, the most common ones being lactoferrin, lactalbumin, transferrin, WAP4-disulfide core domain protein 2, and lysozyme, only 2 proteins are unique to monotremes, the previously discovered EC-AMP and the newly discovered MLP. The MLP protein was purified to study its effect when introduced to microbe cultures. The protein was tested against a staph infection as well as E. facilis, an infection with similar symptoms to strep. The protein was found to decrease bacterial reproduction. As concentration of the protein increased, the growth of bacterial culture size was inhibited, showing a direct correlation between MLP protein and bacterial reproduction. This is depicted in the graphs behind me, staph on the left and E. facilis on the right, where the higher concentrations of MLP from green to blue increasingly reduced no treatment or NT culture. Even the concentration of just 1 microgram had a significant difference compared to the untreated cultures. Overall, this study shows the possibilities of antibacterial treatments and the strange places scientists can find inspiration. The discovery team of the MLP is taking measures and reaching out to medicinal manufacturers and purifiers to test the protein's potence in humans, and hopefully their findings can make strides for medication in the future. That is all for today's story, and as always, you can find more information for these findings on the web.
Hello, and welcome back to Johnson's Biology News Network. I'm your host, Hallie Mitchell. Dyslexia is thought to be one of the most common language-based learning disabilities. It is the most common cause of reading, writing, and spelling difficulties. Of people with reading difficulties, 70 to 80% are likely to have some form of dyslexia. Past studies have reported that action video game, or AVG, training improves not only certain attentional components, but also reading fluency in children with dyslexia. A recent study investigated the shared attentional components of AVG playing and reading by studying whether the visual attention span, a component of visual attention that has previously been linked to both reading development and dyslexia, or VA span, is improved in frequent players of AVGs. The visual processes involved in playing AVGs are similar to that in reading a book. AVGs are known for having a positive effect on various cognitive and perceptual processes as compared to other types of video games. Previous studies showed a positive effect of AVG training on reading in dyslexic children, as it improves visual attention. Temporal and spatial visual attention is enhanced in AVG players. Visual attention span in this context is the amount of letters that can be simultaneously processed in one fixation. A larger VA span relates to faster and more accurate reading. VA span to pseudo-word, or a non-real word that appears to be an actual word, reading is independent of phonological skills in typical readers, but a subset of dyslexic children exhibits a VA span deficit in poor pseudo-word reading despite good phonological skills. Previous studies have shown that VA span is not restricted to verbal stimuli or monodimensional stimuli. Therefore, VA span can be used by players when processing visual stimuli presented by an AVG. So VA span can be a common component between playing AVGs and reading. Scientists conducted an experiment to test their hypothesis. If frequent AVG playing might enlarge the player's VA span, which would help them process larger multi-letter units and consequently improve their reading performance. 36 French fluent adult readers matched on chronological age and reading proficiency composed two groups: frequent AVG players represented by the black line and non-players represented by the dotted line. Participants performed behavioral tasks measuring the VA span and a challenging reading task, reading a briefly presented pseudo-words. AVG players performed better on both tasks, and performance on these tasks was correlated. These results further support the transfer of the attentional benefits of playing AVGs to reading and indicate that the VA span could be a core component mediating this transfer. The correlation between VA span and pseudo-word reading also supports the involvement of VA span even in adult reading. Further studies could combine VA span training with defining features of AVGs in order to build a new generation of remediation software. But in the meantime, go ahead and tell your parents that when you're playing action video games, you're really enhancing your reading performance. Thanks for watching.
Welcome to Johnson's Biology News Network. I'm your host, Nathaniel Lowe. Today's top story involves the fluffiest barnyard animal, the sheep, also known as the Ovis aries. This is not an average Jbnn story. It is a story marred with incredible amount of bioethical controversy, but I will come back to that fact in a moment. Scientists have recently used the technology known as CRISPR/Cas9 to edit specific genes in livestock animals, such as the sheep. CRISPR-Cas9 works by using the Cas9 enzyme, guided by the correctly named guide RNA, which is used to grab specific sections of the DNA by using one end to base pair with the DNA and create cuts in the sequence of nucleic acids using the other end, a DNA cutting enzyme known as a nuclease. Using this technology, they were able to study the effects of the absence of the PDX1 proteins in pancreatic development in sheep. A homozygous deficiency of PDX1 proteins in mice, rats, and pigs leads to results in a lack of pancreatic development. They studied two different stages of sheep development and microinjected them with Cas9 mRNA and PDX1 sgRNA. They used oocytes injected during metaphase 2 of meiosis before being fertilized and zygotes injected 14 hours before fertilization. 40% of zygotes injected had mutations, while 53.8% of oocytes in metaphase 2 injected had mutations, and 20% of zygotes infected had biallelic mutations, and 46.2% of oocytes in metaphase 2 injected had biallelic mutations. Having biallelic mutations means that they have a homozygous deficiency in the PDX1 proteins. Although altogether, they concluded that a lack of PDX1 proteins did not lead to an absence in pancreatic development in sheep. Now let's move on to the good part of the story. Scientists have created the first human-sheep hybrid, which is 99.99% sheep and 0.01% human. That is a whole 1 human cell for every 10,000 sheep cells. By introducing human stem cells into sheep embryos, they created a hybrid human animal known as a chimera. This comes a year after the other major breakthrough in this field, where scientists created a pig-human hybrid, which was 0.001% human. Scientists' end goal in this field is to one day grow human body parts inside of engineered animals to save the lives of those on the waiting list for human organs. However, they still have a ways to go before enough of the sheep is human to grow the organs, which would require the sheep to be at least 1% human. The reason for the bioethical concerns is because of the amount of human the animal is, and because the animal is being created for the sole purpose of having their internal organs harvested. Supporters of this field believe it is necessary to do this to save the lives of those on the organ transplant waiting list. Statistics show that every hour, 6 people in the US are added to the organ list, and every day, 22 people die while waiting for an organ that they desperately need. Unfortunately, the embryos were destroyed by the scientists within 28 days, so we do not know what a human-sheep hybrid would look like. However, we here at Jbnn have spared no expense providing a model of what we believe it would look like. This has been Nathaniel Lowe on Jbnn. See you next time.
Good morning and welcome to Jbnn. This is Deepa Patel here to report today's hard-hitting story. Today's breaking news concerns how flu shot manufacturing forces influenza to mutate. Data from the CDC shows that the vaccine's effectiveness against the H3N2 strain during last year's flu season was a mere 25%. One reason the flu vaccine tends to underperform in years the H3N2 strain circulates has to do with eggs. To produce the vaccines, manufacturers need to grow a lot of flu virus, and they discovered long ago that the flu virus grows extremely effective in eggs. For over 70 years, viruses have been injected into fertilized hen eggs, incubated for several days while they replicate, then harvested from the eggs, killed or inactivated, and purified to go into vaccines. Plus, no other cell system comes close to growing the flu virus as cheaply or as efficiently as eggs. And the industry has invested a lot in the egg production infrastructure, making it hard for manufacturers to transition into more modern technology. According to a new study from scientists at the Scripps Research Institute, the common practice of growing influenza vaccine components in chicken eggs disrupts the major antibody target site on the virus surface, rendering the flu vaccine less effective in humans. In other words, while growing the flu virus for vaccines, the H3N2 mutates to adapt to the eggs, which seems to result in a vaccine mismatch. Scientists use X-ray crystallography to show that when grown in eggs, the H3N2 subtype mutates a key protein to better attach to receptors in bird cells. The H3N2 specifically, there's a mutation called L194P on the virus's hemagglutinin glycoprotein. This mutation disrupts the region on the protein that is commonly recognized by our immune system, leaving the body without protection against the circulating virus. While flu vaccines developed with more modern cell-based and recombinant methods of production have been licensed in the U.S., it is not yet clear if they are more productive against the flu than the egg-based vaccines. And while scientists pursue the holy grail of a universal flu vaccine, they should also try to understand how our current production methods hamper the vaccines we have now. This is Deepa Patel signing off.
Welcome to Johnson's Biology News Network. I'm your host, Hannah Omar, on autism's social defects reversed by an anti-cancer drug. Of all the challenges that come with diagnosis of autism spectrum, the social difficulties are among the most devastating. Currently, there is no treatment for this primary system of autism. New research at University of Buffalo reveals that the first evidence that it may be possible to use a single compound to alleviate the behavioral symptoms of targeting sets of genes involved in the research demonstrated that brief treatment with very low dose of ropidisin, a Food and Drug Administration approved anti-cancer drug, restored social defects in animal models of autism in a sustained manner. The 3-day treatment reversed the social defects in mice deficient in a gene called SHANK3, an important risk factor for autism. The effect lasted for 3 weeks, spanning the juvenile to late adolescent period, a critical development stage for social and communication skills. This is equivalent to several years in humans, suggesting the effects of similar treatment could potentially be long-lasting. Profound and prolonged effect. A small molecule compound that shows a profound and prolonged effect on autism-like social defects. Without obvious side effects, while many currently used compounds for treating a variety of psychiatric diseases have failed to exhibit the therapeutic efficiency for this core symptom of autism. The loss of SHANK3 disrupts neuronal communications by affecting the function of the NMDA, which is N-methyl-D-aspirin precipitator, a critical player in regulating conjunction and emotion, leading to defects in social preference that are common in autism spectrum disorder. In new research, the UB scientists found they could reverse those social defects with very low dose of ropidisin, which they found restores gene expression and function using an epigenetic mechanism where gene changes are caused by influences other than DNA sequences. Human genetic studies have suggested that epigenetic abnormalities likely play a major role in ASD. Many of the mutations in ASD result from chromatin remodeling factors, which are involved in dynamically changing the structure of chromatin, a complex genetic material in the cell nucleus that condenses into chromosomes. The extensive overlap in risk genes for autism and cancer, many of which are chromatin remodeling factors, supports the idea of reversing epigenetic drugs using cancer treatment as targeted treatments for autism. To do so, the team turned to a type of chromatin called histone modifiers, modifying proteins called histones that help organize genetic material in the nucleus so gene expression can be regulated. Since many genes are altered in autism, a histone modifier might be effective. HDAC, a family of histone modifiers that are critically involved in remodeling a chromatin structure and transcriptional regulation of targeted genes. In the autism model, HDAC2 is abnormally high, which makes the chromatin nucleus very tight, preventing the genetic material for assisted transcriptional machinery it needs to be expressed. Once HDAC2 is upregulated, it diminishes genes that should not be suppressed and leads to behavioral changes such as autism-like social defects. But the anti-cancer drug ropidisin, a highly potent HDAC inhibitor, turned down the effects of HDAC2, allowing genes involved in neuronal signaling to be expressed normally. The HDAC inhibitor loosens up the densely packed chromatin so that the transcriptional machinery gains access to the promoter areas of the genes, thus can be expressed. The rescue effect on the gene expression was widespread. The study found that ropidisin restored the majority of more than 200 genes that were expressed in the autism and models. The advantage of being able to adjust a set of genes identified as key autism risk factors may explain the strong and long-lasting efficiency of therapeutic agent for autism. Thanks for watching Johnson's Biology News Network. This is Hannah Omar signing off.